Comparing EEG spectra and event-related potentials, ERPs, with a database can provide important information about brain function. The advantages of these techniques are that they are non-invasive, affordable, and can relate to a large number of scientific studies. Demonstrating these techniques will be Maria Danielsen, a psychology candidate who has worked as a technician in our clinic for about 2 years.
To create a corresponding EEG data file, in the software, click File and New. The subject's card will appear on the screen. After filling in all of the appropriate information, measure the Nasion-Inion distance in the head of the subject. The center of the frontal pole electrodes should fall on a horizontal line 10% of this distance above the Nasion. Apply skin prep gel to the earlobes and fill the ear electrode cups with conductive electrogel. Attach the ear electrodes and place the cap symmetrically on the head, with the frontal pole electrodes placed in the center of the frontal pole. Pull the cap down as far as possible to secure it close to the head, and place the waistband around the chest. Fasten the push buttons connected to the cap to this band to prevent the cap and electrodes from moving during testing, and connect the cap cable and ear cables to the amplifier. Next, click on the Win EEG menu Control of Impedance icon, and fill the syringe with